Today we are going to be talking about our muscle cells and how muscle cells look on a cellular level. These people just finished a marathon. They have used their muscles for so long they must think they look like jelly. But really, muscle cells are composed of many different types of filaments that allow them to function properly. There are three main components that divide the muscle cell: myofibril, sarcomeres, and Z-lines. We are going to now discuss each of these components in detail. Myofibril and long strands made up of sarcomeres, which we will discuss soon. Interestingly, the nucleus of these cells sticks out of the myofibril groupings and is not internal like other cells. Now we will discuss sarcomeres. Sarcomeres are little section that comes together to make up the myofibril. Think of tiles on the floor. It is all floor, but the tiles are individuals. This is what sarcomeres are to myofibril. Sarcomeres can further be divided into two categories that make it up: F-actin, which is a thin filament, and myosin, which is a thick filament. F-actin is a thin filament. It does move. Myosin, which we will discuss in the next section, walks on F-actin, which will cause muscle movement. Myosin is a thick filament. Myosin heads and bundled together at two opposing ends of the sarcomere with the tails meeting in the middle. The myosin walks on the F-actin in order to facilitate muscle movement. That's a good question. This is where the final component of the muscle cell fiber comes in, the Z-line. The Z-line is a thick line that divides the sarcomeres. They allow the sectioning of muscle fibers, which helps control our detailed movement abilities.
So our muscles are not actually made of jelly. Just a lot of different components that are all really hurting right now.
I guess not. Thanks for listening, and I hope you now understand your muscles even more.